My name is Danielle Slaughter, and today I'll be presenting our work studying placental hypoxia and mitochondrial damage in preeclampsia. Preeclampsia is a pregnancy complication that affects 5 to 8% of pregnancies worldwide. It's believed to begin with an issue as the placenta implants into the uterus that results in poor blood flow and a hypoxic or ischemic placental environment. Through unknown mechanisms, this results in preeclampsia, including maternal systemic endothelial damage, hypertension, and proteinuria or other end organ damage. Delivery of the baby is the only available cure. So based on many background studies, we proposed a novel mechanism for preeclampsia. Our hypothesis is that placental hypoxia leads to increased reactive oxygen species, which go on to damage the mitochondrial DNA within the placenta and release this DNA as fragments into the placental microvasculature. Here they bind to toll-like receptor 9 on endothelial cells and lead to activation of the innate immune response, including inflammation, endothelial dysfunction, and preeclampsia. For this work, we looked at the first step of this mechanism and wanted to study, does placental hypoxia lead to increased reactive oxygen species within placental endothelial cells? To test this hypothesis, we used a human microvascular endothelial cell line called HMEC1. Cells were grown and split onto multiple plates, followed by incubation for 24 hours to allow the cells to adhere. At this time point, the cells were then moved to either a 1% oxygen incubator, simulating a placental hypoxic environment, or a 6% oxygen incubator simulating a placental normoxic environment. These conditions were previously established by the placental research community. After 24 hours in either the hypoxic or normoxic environment, we then applied a mitoxox dye solution. Mitoxox is specific to reactive oxygen species within the mitochondria and upon interaction will fluoresce bright red. We simultaneously applied a hoax dye solution, as hoax dye is specific to cell nuclei and upon interaction will fluoresce bright blue. This allowed us to control for the total number of cells present. The second assay that we ran is propidium iodide assay. Propidium iodide can only enter the cell once it is dead, and upon doing so will interact with the DNA and fluoresce at a specific wavelength. This allowed us to compare cell death between the two groups. Fluorescence was measured using a spectrophotometer or visualized under a microscope using fluorescence microscopy. Figure 3 displays the results from the mitoxox fluorescence microscopy. HMEC1 cells incubated in hypoxic conditions are on the left, labeled as image H. Cells incubated in normoxic conditions are on the right, labeled as image N. Blue fluorescence indicates cell nuclei. Gross inspection of the hypoxic cells show areas of bright red fluorescence indicating presence of mitochondrial reactive oxygen species, as seen within these circles here. While several spots of red fluorescence can be seen in the normoxic cells, as indicated by these arrows, overall, fluorescence appears brighter and greater in number within the hypoxic cells. This indicates a greater amount of mitochondrial reactive oxygen species within the hypoxic cells as compared to normoxic. Figure 4 displays the spectrophotometer results. 4 samples were studied for each group. Figure 4a on the left shows the mitoxox fluorescence after controlling for the total number of cells present as determined by hoax fluorescence. The mitoxox fluorescence for the hypoxic cells was not significantly different than the normoxic cells, and this indicates that total mitochondrial reactive oxygen species were not different between these groups. Figure 4b on the right shows the propidium iodide fluorescence. This was also not significantly different between the hypoxic and normoxic groups, and indicates no difference in cell death between these two treatment conditions. So while spectrophotometry did not show a difference in mitochondrial reactive oxygen species between the hypoxic and normoxic HMEC1 cells, we did observe areas of increased mitochondrial reactive oxygen species fluorescence in the hypoxic cells upon gross inspection under the microscope. These preliminary findings lend some support to the first step in our proposed novel mechanism of preeclampsia. Study strengths include the use of the HMEC1 cell line, which has been shown to retain the key characteristics of normal human microvascular endothelial cells. However, preeclampsia is a complex disease, and use of a single cell line is also a limitation. Future work will repeat these experiments and determine if our observation under the microscope is significant. Additional studies will include the effect of hypoxia in placental trophoblast cell lines, as well as human placental samples. We believe that elucidation of a mechanism for preeclampsia is important, as it may one day lead to a therapeutic option other than delivery. So in conclusion, our findings suggest that hypoxia may increase mitochondrial reactive oxygen species within human placental endothelial cells. I would like to thank the Medical College of Wisconsin Cardiovascular Center and the NIH for funding this work. Thank you for your time.